So this is 2015 Scape Biology paper unit 1, paper 1. The first question here, it looks like it has been repeated multiple times. So waterless is a rosette-like plant found floating in ponds and natural waterways. Which of the following properties of water assists the plant to float? What will be the answer?
A.
A. Water's high cohesion
A.
keeps so it keeps the water molecules together, creating a high surface tension, which is relatively hard to break. Number 2, which of the following combinations correctly describe the structure of molecules 1 and molecules 2?
C.
C. Right, so 1 is a protein, 2 is probably a dimer of cellulose. Right, looks like it. 3. Sucrose is a good transport molecule as seen in phloem tissue because sucrose is a. Is it because it's a disaccharide? Is it because it's very reactive, or it's because it's very
Is
soluble?
it C?
Sucrose is very reactive? I
Sir
don't
D?
think so. It'd be more likely D. Because it wouldn't be so because it's a disaccharide. There are tons of more disaccharides, right? It wouldn't be so because it's a disaccharide made of glucose and galactose, because it isn't. What's a disaccharide called when it's made of glucose and galactose?
Sir lactose.
Lactose. So it's not it. So it's very reactive molecule chemically and therefore combines easily with water. Why would it be good at transport? If it combines very easily with water, will probably all combine with water and all reduce, right, as a reducing sugar, if it is a reducing sugar, right, before reaching a storage organ. So it's important that it's not very reactive. So not very reactive would be it. So it eliminates C there. So very soluble molecule and can therefore be moved efficiently in high concentrations. So that would be the best thing there. Here now, which of the following biological molecules forms the main component of the cell membrane in animals?
D.
The phospholipid. Alright. 5. Which of the following molecules has both hydrogen and disulfide bonds? Which would it be? Remember, I can't see the chat unless I move my screen. Let me check in the chat real quick. Looks like someone says B. What would be the answer here? What's ATP? Could somebody tell me what that is?
Adenosine triphosphate.
The triphosphate, right? So for adenosine triphosphate specifically, it will have covalent bonds, right? Whether it is hydrogen bonds or disulfide bonds, right? It may be a bit tricky for persons who haven't looked at the in-depth molecule, right, before you need to, right, to do. But do you think you find sulfide or sulfur in ATP? I don't think so. I don't think we'll find sulfur
No, sir.
in ATP. For DNA, do we find sulfur there? We'd have hydrogen bonds, but not disulfide bridges, right? And for triglycerides, right? For triglycerides, I don't think so. What is insulin?
A protein.
That's a question mark or a full stop? It's a protein.
It's a protein.
Alright, it's a protein. So it'd be most definitely the one with the tertiary structure or quaternary structure, right? So I would like somebody to put in the chat whether or not, insulin as a tertiary or quaternary structure. Alright, after they find it. Okay, it's a protein. 5. 6. Which of the following combinations
Sir,
shows...
could you repeat what you said?
Okay, about number 5.
Yes, sir.
So we're saying that insulin would be the most likely answer because it is a protein. Is it not a protein? Is insulin not a protein?
Yes.
Okay. Somebody say no down here? Okay. So it will not be a protein. Well, it will be a protein. So it will most likely have the disulfide and the hydrogen bonds because of protein structure, whether it be the tertiary or quaternary. So it's asking if anybody could find out whether or not insulin is tertiary or quaternary in structure, and put it in the chat. So it's because it's a protein. DNA wouldn't have sulfur, ATP wouldn't have sulfur, triglycerides wouldn't have sulfur, right? For the most part. The general depiction of the molecules we see don't have sulfur there, so it wouldn't have disulfide bonds. Alright. 6. Which of the following combination shows the correct sequence of solutions in ascending order from least amount of reducing sugar to the most amount of reducing sugar?
A.
A. So we're seeing 1, and this is a repeated question. Solution
Sir,
4.
B.
Alright. So it would be B. So it was a repeated question. Hold on, let me let in all these questions. Okay, so let's go. Number 7. Which of the following is not likely to be seen in an electron micrograph of a plant cell?
C.
C.
C. Centrioles are not related or not associated with that. The actual diameter of a stem is 10 millimeters. What is the magnification of the drawing?
A.
A.
What is magnification?
Image size of actual.
So the size of the image over the actual size. Alright, so we have the actual diameter and then we have the image size. So we know what to do with that to get approximately 5. Right? A. So what we're gonna do, image over actual. So the image is 50 over actual, which is what? 10. So that would give us what? 10 goes into 55 times. Alright. So number 9. Hold on. I have to let in persons every other minute. Which of the following combinations correctly identify structure, typical of an eukaryotic cell?
B. B.
B, beautiful. We have to remember that the ribosomes for eukaryotic cells or RADS. Why is this not writing? It looks terrible. ADS. Alright. 10. Bobby is given a tropical freshwater fish for his birthday, experimenting with the fish. He places it in seawater and it dies. Which of the following statements most likely explains the reason for its death?
B.
B. B.
Biology papers tend to repeat a lot. I have no idea why. They should mix it up this year. Should they? Let's see. 11. Which of the following correctly describes the importance of cholesterol in the cell membrane?
D.
D.
Alright. Remember it adds to fluidity. So whether or not they ask for, well, they give fluidity or flexibility, then you'll use those. Alright. So 12. A plant cell is placed in a solution A which has lower, not higher, but lower water potential than the cell contents. Which of the following combinations shows the most likely change in the cell and cause of the change?
Sorry.
A.
Sir, C. C.
See? It has lower water potential. So we have a cell, right? That's a general water. Let's call the cell water potential 0, even though that's not a thing. Just using it for explanation, right? So we're saying that it has a lower water potential. That means it would be more negative or more positive. Which one?
More negative.
More negative. So let's say the solution outside is negative 100. Right? What does water potential do to the cell? So if outside has less water potential than inside, what type of solution is that? Hypertonic, isotonic, hypotonic,
Hypertonic.
Hypertonic.
hypertonic. And what happens within hypertonic solutions? Water flows in or out?
Out of the cell into the solution.
Okay. So water will flow out of the cell into solution. Alright. And if water flows out, it makes the cell flaccid. There's another question like this where it says more water potential or higher water potential. What would that answer be?
Sir, can you repeat that?
Okay, I was saying that there's another interpretation of the question where it says higher water potential. So what would the answer be? People are
Sir,
saying
A.
Would have been
A.
B.
B.
The water is what moves, not the solution. Well,
Beautiful. Alright, so it would have been B. It would have been B. So we have to look at that. Look carefully at what is moving, guys. Which of the following structures A, B, C, or D is important for cell-to-cell recognition?
D.
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 D would be the best answer. Even if you didn't have, well, even if you wouldn't have completed this section of biology, right, in depth, you would have known the general structures, such as the peripheral proteins and the transmembrane proteins, and that wouldn't have anything to do with communication. So it must be the one that looks like an antenna. Well, I'm just saying that, alright? So it must be the one that is odd. Alright, so this is the next repeated question. Pepsin is an enzyme found in the human alimentary canal. Where in the alimentary canal?
The stomach.
Right, the stomach. Alright, so which of the following, well, which of the above accurately, well, activity curves represents the activity of pepsin?
A.
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 Yeah, alright. Hold on. Okay, so it would be A. Wait, give me one moment there. Give me one moment, guys. This thing is 15. According to the endosymbiotic theory, organelles such as chloroplast and mitochondria have evolved from prokaryotic organisms. Which of the following is not a line of evidence in support of this theory?
A.
A.
Selection A. Alright. Many bacteria are similar in structure to. Okay. So B would support it because they both contain DNA. C would support it because they are similar size to bacterium. And D will support it because the photosynthetic bacteria are largely similar to chloroplasts, right? Well, bacteria aren't similar to eukaryotic nuclei. That doesn't make sense to me. 16. Which of the following is found in both DNA and transfer RNA?
C.
A.
You see? Okay. Give me a moment. I have to like jump in here every other second. I'm gonna join the meeting with my phone so I could just accept them from there. 17. In protein synthesis, which two of the molecules illustrated below are involved in the process of transcription? So which are involved? So which two are involved in transcription? Are we getting cold feet? What's transcription? What's transcription? Anyone tell me that?
It's a process where okay.
I'm not hearing anything. It's process where okay. That's what I heard.
It is it is the process where code from DNA or genetic code from DNA is copied onto an mRNA molecule.
Okay, that's interesting. So it's copied. So for transcription, would we agree that the double helix has already been separated? The hydrogen bonds have already been broken. Okay, looks like I'm going to have to look in the chat because nobody else.
Yes.
Yeah, okay. So do you think 4 will be involved?
No.
Okay, so we're saying 4 is not involved. So B and D are out. Look how quickly it's gone. Now what is 1?
It is a nucleic acid.
Looks like a nucleic acid? Alright. What is 3 though? Do you think 3 will be involved? Let's look at the more likely options. Do you think 3 will be involved?
Yeah, it is look like a half strand of DNA.
So it looks like it, right? So everything here is not really definitive, but it looks like it, right? So we're saying that 4 is not involved and 3 is involved. That means that probably C would be the answer, based off of the process of elimination. 1 looks like ATP to me, right? It looks oddly like ATP to me, right? It just looked like it. Alright. So we're saying that 3 must be involved. This question is a bit odd, right? From my perspective. But by the method of elimination, we say 4 is not supposed to be there and 3 has to be there. That means that C is the answer, even if everything else isn't as conclusive as we'd like it to be. Alright. So number 18, which of the following amino acid sequences would be produced from information coded on the DNA strand shown below? Would be tyrosine, then arginine, then leucine. Alright. Okay.
So it would be B.
Yes, it will be B. So we know we just create the RNA strand for it and then match it with the different proteins. 19, which of the following is not a feature of the genetic code? Okay, so we're saying that D is the answer. So let's look at this. Each species has its own set of unique triplet codons. The central dogma of genetics speaks to the fact that DNA codes for RNA and RNA translates it into proteins, right? No matter the organism, isn't it so? Would be used regardless of organism, right? So that's the genetic code. So I would believe that D would not be a feature. Someone asked what was 17. 17, we put C. 20, let's look at that. The following diagram shows the structure of a tRNA molecule. Which letter A, B, C, or D indicates the position on the molecule where the amino acid would be attached?
That'll be A.
A, beautiful. Alright, 21. Which of the following features are evident during mitosis? Mitosis.
So B.
B, okay, so we're saying 1 and 2. let's look. Two daughter nuclei containing identical sets of chromosomes. Alright, interesting. And we're saying what? 4 homologous chromosomes do not associate. That's what we're saying? Interesting. Let's look at 2. Homologous chromosomes come together and crossing over occurs. That's genetic variation there. That doesn't happen in mitosis. No. In 3, 4 daughter. No. We use 2. So it has to be 1 and 4. B is the answer. Great. So let's look at this now. 22. Which of the following is not true of most homologous chromosomes?
B.
This question has been repeated for a long while now. Let's look at this. Okay, we have 2 regions. So if crossing over takes place in region 1, which of the following alleles will segregate at meiosis 1? So remember, those are the centromeres, you know. Right? So those are the centromeres. So what we're depicting here is that we have, right, the chromosome and we have the centromere here. So we have large B, and then we have small b, and then we have large A and small a. So in crossing over, which alleles will segregate in meiosis 1? Oh, I'm just seeing some. Oh, I wrote it wrong, you know. Hold on. I wrote it wrong. It should be large A down here, on this loci here. And then we have small b. Alright, so what was going to be? You're going with A. Somebody going along with A. So within crossing over, what happens to the chromosomes? Someone talk to me.
The non-sister chromatids interact with each other and the alleles switch.
The alleles will switch, they cross over, alright? So, in meiosis 1, right, would it just be chromatids or the entire chromosomes?
Repeat that.
We're asking, right, for meiosis 1, right, is it just sister chromatids interacting or entire chromosomes interacting?
The
Entire
entire chromosome.
Entire chromosome. Alright. So we have to know what is switching over, what is crossing over here. Alright, which ones will be crossing over? Or which ones will be segregated, right? So people are saying C, well, A, I mean. We're saying that A is the best answer. Why do we say A? Why do we say A? Okay, it looks like we're gonna have a while here. Okay, so let's highlight this question here. Set A. Alright, so the fur color in mice is controlled by a pair of genes occupying different loci. Large W represents the presence of colors, small w represents albino, right? So the type of brown, well, sorry, the allele for brown fur is large A and black fur is small a. The genotype expressed here is brown, black,
C.
albino.
C.
Okay, albino. 25. What is the phenotypic ratio of F2, right, the second generation, from a cross between two first generations with the genotype expected here? We did this entire question before, right? What was the answer?
C.
Thursday.
C was the answer?
Yes, sir.
Okay. We did the question before now, so you're sure C is the answer? It would be 9 brown. Yeah. Alright. So 26. A possible benefit of producing transgenic plants is?
B.
Beautiful.
C.
27. In the production of recombinant DNA, restriction enzymes are used to cut double-strand DNA molecules to form fragments. Which bonds are cut by these enzymes? Which bonds are cut?
Hydrogen bonds.
Why? Why would it be that? If we have double helix of DNA, alright? Right, we have the double helix of DNA. Hydrogen bonds connect vertically in this diagram, right? So in order to cut hydrogen bonds, you'd have to go that way with helicase, right? But what restriction enzymes tend to do is cut this way, right? They usually cut in a shape like that, right? So they'll have to break phosphate bonds, isn't it so? Because they're literally cutting the entire DNA strand, you know. So they're not necessarily cutting the hydrogen strand. They're keeping the bases are still combined, right? Except for the sticky regions, but they're cutting through the entire DNA, right? So restriction enzyme will most likely focus on the phosphate backbone, which you'll have to cut through the backbone. You'll have to cut through the backbone, right? So that would be the most likely answer. Following?
Yes, sir.
All right. You have to cut through the sugar phosphate background. 28. Which of the following is not, right, one of Darwin's observations about natural selection?
D.
A. There is a variation among
B.
individuals.
D.
Okay, B. But that's not true of their observation. 29. Mutations can have varying effects on an organism depending on when it occurs. The occurrence of a mutation has the greatest effect during.
Sir,
Okay, go ahead.
could we go back to 28, please?
Okay.
Why did we say B again?
Okay, so let's look at all of them then. There is a variation among individuals of a given species. Alright? Would that not be Darwinian observation?
Yes, sir.
Because he looks at the differences within the species and then look at the differences outside of species, saying that with great variation, speciation can occur. With C, natural populations tend to remain stable in size over long periods.
Yes, sir.
So we're looking at the stability of populations due to variations that will prevent genetic diseases, right, and environmental upheavals. D, populations consist of those individuals best adapted, survival of the fittest. This is one of the central concepts of Darwin's theory of natural selection, but it's not. Oh, go ahead.
I was saying that Darwin also said that, well, he observed that organisms over reproduce, so wouldn't that thing?
Okay, let's see. So you're saying that they over reproduce?
Yeah.
So is it specifically in keeping with the purpose of replacing parents, or just that they over reproduce?
Well,
Because this is saying that the organism produce more offspring than needed to replace the parents. So you're saying that it's over reproducing. All right. So which one is not? Well, about natural selection specifically, right? I think it's speaking about natural selection. What is natural selection? How Darwin observed a lot of things that contributed to a lot of things in natural history and sciences, you know. But what is natural selection? And what contributes the most to natural selection? Or the least in this case? What is natural selection? Come on, man. What is natural selection?
A process in which organisms that are best adapted to their environment survive, while those less adapted will.
Okay, simplified but truthful. Alright. So we're looking at natural selection specifically, about natural selection specifically, right? So we're looking at the variations, right? So usually we're going to look at the variations generally for natural selection, right? And that populations remain stable, right? Natural populations tend to remain stable. So when he's talking about natural selection, well, natural population, he's saying that natural populations tend to have, you know, a natural variety, you know, of different genes and different species, right? That will give stability to the environment through biodiversity in some type of cases. Give me a second. Let me see. But can't the population consist of both species with selective advantages and disadvantages? Just that those who have the selective advantages would be small, right? That's what he's saying about the natural populations. Will have a stable, will be stable in size over time, over longer periods, right? Because if the populations are not natural, right, if they are filled with completely advantageous organisms, right, then they'll expand over time, right? They won't remain stable. They'll have exponential growth, right? If they are all disadvantageous, it will decrease over time, leading towards, you know, endangered species and issues of extinction. So if they have natural variations within natural populations, it will remain stable over time. It should remain stable over time. For organisms producing more than they need from their parents, right? I don't think that that specifically speaks to his theory of natural selection, right? It is something that he observed, but does it speak to the fact that genetic differences, right, occur within, you know, sexual or reproducing organisms, right, to produce organisms that may or may not be better adapted for the next, you know, for their environment, right, or for their niche. I don't think it specifically speaks to that, but what would specifically speak to it is that the organisms produced, right, genetic, will have very genetically from their parents, and only the ones that have the best advantages will survive. Go ahead. Somebody's saying something.
So sir, could you just explain what's the difference between what he observed and what he concluded, or they're just the same?
Okay, it wouldn't be a difference between what you observed and what you concluded, right? Well, observations are different from conclusions specifically, right? So we're going to have to look at Darwinian observations then. Probably we're going to have to look at that, right? But it could be a... Okay, why did my brain just freeze? Okay, so it could be an issue in which, right, he observed that the organisms produce more offspring than needed to replace their parents, right? But what we see here is that even though they create large populations, right, or produce large amount of offspring, right, the natural populations still remain stable. Why isn't the natural populations of these species skyrocketing out of control? This is probably because of the variation, right, and mutations. and the fact that some of the species, right, will have a better adaptation for their environment, and some of the species will have worse adaptation for their environment. Note that usually species who create a tons of organisms, right, multiple of them usually die off, right? It is a way of securing that some go on to the next generation, right? Some fishes create hundreds of organisms, right? 50 will survive, right? So you're looking at the fact that only the best will survive, so it links into D. But it's not the mere fact they produce a lot of organisms, right, speak to natural selection, right? It's not just that specifically. I wouldn't say that specifically will do it. But let me put an asterisk to this question. It shows that what? So my argument is that statement alone wouldn't have contributed to the groundbreaking theory of natural selection alone, right? It would be what is happening in the genetics or the variations, right, of these organisms, right? Because despite, right, the organisms producing more offspring than need to replace the parents, right, that is something that is observed, right? But why haven't it created an unstable population? Right, that doesn't specifically speak to natural selection. Natural selection speaks to variation and adaptability. Creating more offspring than need to replace parents isn't really what I would call that natural selection per se, right? But we could argue that, right? I could just leave an asterisk there. Let's complete the paper and then we could argue that. 29. Mutations can have varying effects on the organism depending on when it occurs. The occurrence of a mutation has the greatest effect during
C.
DNA replication.
I think it's A.
Is the greatest effect. DNA. Okay, mutations won't generally have any if. Okay, so what we're saying here, right? For the effect, mutations are created mostly in replication, when replication goes wrong or when transcription goes wrong, right? So mutations will occur there, you know, right? But the greatest effect of mutations, right, the greatest effect of the genotype of the organism is displayed in its phenotypic traits, right? So what I would believe would be stated here is that the greatest effect of the mutation will be, right, the outcome of the protein that will be created after translation. So for example, sickle cell can live, well, sickle cell, its variation can be present within DNA replication, right, and RNA synthesis, well, let's say transcription, right? It can be present, right? But it won't technically have any effect on the organism unless it starts to translate into proteins that will create a sickle shape out of the red blood cells, out of the erythrocytes. So I would probably argue here that the greatest effect it would have on an organism is through translation, as it creates proteins that will adversely affect the functioning of the overall organism, more so than it existing within DNA replication. Within DNA replication, it will show the greatest variation or the greatest chance of mutation can occur, right? But it will actually affect the organism when these proteins are being created and it realizes that these proteins, right, are disadvantageous to the original purpose that it must fulfill. So I would believe that A would be the best answer. Do we have variations to this answer? And any explanations for it? Going once, going twice.
I was thinking that it would be DNA replication, because if you have the wrong codons, then you can get the wrong protein, which can then lead to different outcomes. So I would say C.
Okay. All right. You're saying, wait, you're saying D? Did you say D? Or
C,
C?
C.
Oh, okay. So let's read the question. Mutations can have varying effects on an organism, right? Notice it will have no effect on the organism unless it's translated, depending on when it occurs, right? So it has to be translated for the effect to take place. It will not affect the organism within DNA replication, right? Or transcription, right? It has to affect the organism after the proteins are translated and the proteins are seen to be null and void, right? The occurrence of a mutation has the greatest effect. The occurrence of the mutation occurs within DNA replication and then has its effect in translation. That's my argument. Because it occurs within DNA replication. It does occur. It can occur within transcription as well. It can occur through, radio waves or infrared radiation, right, affecting the genetic material or the genome of the organism. It occurs there, but I would believe that we see the greatest effect in translation. That's just what I'm saying. So let's put an asterisk here. Another issue with this question. So do we still agree it's C? Well, are we still split between A and C?
So what is like the answer? Is it A or C?
I would say that my answer would be A, but some persons would say C.
I saw in a solution that it was C.
A
I
solution?
did this in an exam, for my mock exam, and I wrote
Mhm.
A and I got it wrong. My teacher said it was C.
Okay. All right. Okay. 30. Which type of isolation plays the most significant role in allopathic speciation?
D.
A.
31. The horticulturalist has bred hibiscus varieties which bears violet flowers. Which of the following propagation would be most appropriate for rapid production of a larger number of clonal plants? What would it be?
D.
D.
32. Which of the following occur in asexual reproduction only?
A.
So offsprings are genetically the same and gametes most never involved. Okay. 33. Which of the following terms is the most appropriate to describe a plant in which the anthers of the flower mature before the stigma?
A.
A.
Right? Which one of these are? Okay, this repeated. So you would have to be the answer for that. Okay, so I'm not sure if it's fine to save time by just answering the questions I have been continuously repeating for a long while now. But yeah. Right, these have been repeating for a good while now.
Sir, hold on. What is 36?
Oh, 36. A.
With 23 BC.
23. Let's check on 23. 23? You said?
Yeah.
Okay, going straight back up to 23. 23. Would it be C? Why would you say that? Why would you say that? Okay, let's try to finish the paper, right? And then we'll discuss and make changes after. Alright? So let's have a look at this, right? So where this fertilization takes place, it takes place in the fallopian tube. Right, for 40 during spermatogenesis in a human, the first cell to divide mitotically, right, are which one?
C.
C, primary spermatocyte. What would it be? Are we sure it's a primary spermatocyte? Are we sure?
Yes.
Alright. Hold on. So spermatogonia would be mitosis. Then after it's there now, it will be the primary spermatocyte. Alright. And that would... Okay, so what would be the answer for this? Has been repeating for a good while now. So this would have been B. Alright. So those are pituitary hormones, and then estrogen and progesterone are the ovarian hormones. Looking at this one, the sample of a woman's urine has been tested, right, at a laboratory, and human chorionic gonadotropin is detected. This indicates that
D.
D.
fermentation has occurred. 43, the hormone which stimulates the development of the endometrium lining is
A.
alright. The correct sequence of events here. What is the correct sequence of events?
B.
B. Alright.
Excuse me, sir. What was the answer for question 41 again?
41. B here.
Okay, thank you.
Alright. So let's see. So we're saying that the inner mass, cell mass forms. Sounds like the blastula to me. Chorionic villi grow, then the maternal blood is formed, the allantois fuses with the chorion. Alright. Seems fine. 45, during the development of the fetus in the womb, the uterine walls consists of the muscle of the uterus wall and the
A.
endometrium. Alright.